everyone, my name is Sean Lee from Institute for Composite. Here's a question to you guys. Did you eat hamburger this week? Yes. Well, I bet some of you might have seen a picture like this before. A hamburger is often served with 2 slices of bun inside with cheese, lettuce, tomato, onions, mayonnaise, and most important thing, the meat. 100% pure pork, grilled juicy chicken, and the beef directly from Australia with a pinch of pepper and salt. We all emphasize on how great the meat is. But now, if someone tells you that the lettuce and hamburger was grown in the farmland with automatic monitoring system, the farmers can monitor the condition of every plant in one specific area, and at the same time predict the number of pests that will soon appear and harm in the near future. Also, in the farmland, the system sprays a kind of biological pesticide that is perfectly no harm to humans, and the farmers can take care of every slice of vegetables when they are taking a duck tour in the Charlotte River only by nap. When you change your order from double cheese into double lettuce, it sounds great, right?
Yes.
Yes.
In 2016 Institute for Formosa, there are 2 groups of people with 2 radically opposed worldviews on agriculture. One is biology, the other is engineering. By the interdisciplinary cooperation, we would like to introduce a brand new system that revolutionizes traditional agriculture. Let the journey begin with the biologist, Vivian.
Life is just a code, and we are using it for practical application. The inspiration comes from one of the most successful predators on Earth, spiders. Webs, fangs, and venoms. Spiders are a fascinating creature with genuine predator skills. Here we provide a new remedy for pest control with the extraction of spider venom DNA sequence, that is, peptide. Peptide is a new word coming from 2 separate words, pan and peptide. Pan is the god of shepherd and nature in ancient Greek mythology, and peptide indicates the components of peptide. Peptide derives its toxic effect from spider venoms, and spider venoms are a mixture of more than 5,000 toxic peptides that act as neurotoxins. But if we choose the toxins with our eyes and hands, it will take months. But if we use the software we designed, it only needs a few minutes with a few clicks. The software will process data from several big databases, including UniProt and the Ragnar server. Then, different conditions were set to filter nearly 800 toxic peptides, including the species of targeted pests, toxicity mechanisms, and the numbers of disulfide bonds. Then, through blast, grouping, and cobalt processing, we got 3 largest toxin families. Then we chose the representative of these 3 families. They are HV1A, omega-hexatoxin HV1A, SF1A, mu-suggestory toxin SF1A, and OAIP, orally active insecticidal peptides. These 3 toxins bear a structural resemblance called ICK, inhibitor cysteine nut. ICK is a nut-like structure. The high density of disulfide bonds in ICK make peptide more stable in temperature, pH solution, and protease. It blocks the voltage-gated ion channels of insect that causing paralysis and finally reduced appetite. But here comes the question. How does peptide get to the nervous system of insect? By ingestion. So we also designed a fusion protein with the addition of snowdrop lectin. Lectins are commonly found in plants, and they have great affinity to glycoprotein. Snowdrop lectin binds to the glycoprotein on the surface of the epithelial cell in the alimentary canal. Therefore, snowdrop lectin is a carrier that transports peptide across the epithelial cell by transcytosis. Peptide is a biorepellent, and we think of peptide as an impeccable alternative of chemical pesticide, because peptide possesses several fascinating features. First, it is safe to humans. Due to the significant structural difference between insects and mammals' voltage-gated ion channels, peptide will not block the ion channels of humans. Second, it is biodegradable. Peptide will degrade into amino acids in the natural surrounding. Third, it is eco-friendly. The 3 toxins we chose to target a specific species of insect, including Lepidopterans, Dipterans, Coleopterans, and Orthopterans. Thus, it will not harm other organisms and pollinators like bees. Safe, biodegradable, and eco-friendly. These 3 characteristics guarantee the sustainability of agriculture and safeguard human health. Next, let's welcome Angel to tell us more about pentie.
In the biobrick construction, there are two types of biobrick, with a strong T7 promoter and histech for protein purification. One is a single tyrosine peptide of the three chosen tyrosines, H1A, SF1A, and OAIP respectively. The other is the addition of snowdrop lectin. And we use E. coli PO21 was a target strain to express pentie. This strain of E. coli is powerful at expressing proteins that contain multiple disulfide bonds. To prove that our biobrick truly works, we have done several experiments. First, the cloning result. We did PCR to ensure that the genes are cloned into E. coli. We may select the two types of toxins without or with lectin fall in the correct ranges. Second, we want to make sure that E. coli successfully express pentie. The SDS page results show that the toxins are falling into the correct ranges comparably to markers. Third, we want to test if pentie performs any effect on the pest insects. So we use tobacco carbon larvae as testes for testing the toxic effect of pentie. After expressing proteins, we apply the samples with purified products onto the leaf discs. Primarily, we want to test if the larvae have any preference on the pentie plant leaves or on the original ones. In the video, caterpillars become elevated and they prefer to eat native control leaves. Based on this observation, we estimated that pentie has the repellent effect. To further discover the qualitative effect of pentie, we apply the pentie-expressed sonicated E. coli lysate with 3 different concentrations onto the leaf discs as pretest. Next, we calculate the remaining leaf area with software, and this is the result. Take H1A as an example. Compared to the positive control, Bacillus thuringiensis, a widely used biological pesticide, H1A even performed the same efficacy. As the concentration of H1A increased, the remaining leaf area gradually increased. That means H1A has the dose response. Besides, we chose H1A to quantify its efficacy. In this experiment, we also changed the original linker with a longer Jens linker that allows the 2 domains for proper folding and tested if H1A will have the improved toxicity effect. So we applied H1A lectin, and H1A lectin with Jens linker onto the leaf discs. Here are the results. The graph shows that H1A lectin compared with H1A has significant difference. And H1A lectin has better toxicity effect. And the toxicity effect of H1A lectin with GS linker is the best one. This perfectly supports our hypothesis. Also, SF1A and OAIP show a similar tendency. In the 3 feeding assays, we had 3 conclusions. First, pentie performs its toxicity as a repellent in vivo. It is an interesting finding that goes beyond our original hypothesis. Second, the fusion proteins of the 3 toxins with snowdrop lectin enhance the repelling effect. And third, with the elongation of linker, the improved fusion protein has the best repelling effect. The toxicity effect of pentie did not kill the pests, but it did perform a reduced appetite and reduced mobility after ingestion. It can be an effective biological repellent that lowers the artificial impact on the environment and at the same time retains pest control. Genetic information makes us easy to express pentie that fight against the pest. As engineer, Oliver, can you share about your opinion of the practice of pentie in the farmland to the audience?
In my view, pentie should be sprayed onto the farmland in the correct and accurate timing automatically. To achieve this, we create a detection device. It can monitor the conditions in the farmland, including temperature, soil moisture, humidity, and the number of pests. In the detection device, we use P-band to attract pests. And we also designed a larvae prediction modeling that can accurate adult moth to predict how many larvae are in the farmland now. Here along the entrance wall, there is infrared ray, also used to count the number of insects and close the lid in case a pest gets out of the cabinet. Furthermore, our device is installed with Arduino that processes the information and uploads the data to the app and cloud. But why we describe it as a totally automatic system? The answer is a function of IoT talk systems. IoT talk, a more modulated and more user-friendly version of IoT, is like a bridge between the hardware in the farmland and the farmers. Supposing that we were on a big farm, and the farm is divided into several parts. Because our device has accurate detection range, about 2,000 square meters, we put one detection device in each part. From one of the areas, which is the spraying value, we can activate the spur of that region at once. All the process is done with IoT talk through Wi-Fi. The other important features of IoT talk is a function of customizing. Different farmlands have different environments, therefore different conditions. The cloud can record all the data in the farmland. That means it builds a calendar database for every farmer. It is quantifying the everyday conditions of the farmland into annual calendar for future prediction. And most importantly, we are the first team that integrates IoT talk system into synthetic biology.
And what's the next step after the device detecting the conditions in the farmland?
Our device is coupled with Pentax spraying system. Now let's welcome Tony to demonstrate how our device works.
Okay, let me show you how it works. Imagine that we were farmers, and here is our farmland. The only thing we need is a smartphone and a detector. On the screen is the app that displays the data from the detector. If we put the detector into the farmland, the data will be transmitted to the app. But that's not all. This is, beyond the doubt, the most precise detection. If a mouse flies into the device, the infrared ray in the channel will detect the moving insects, and the number of paths will be shown on our cell phone. The data will be sent to a routing prediction modeling in IoT talk system for deciding when to spray Penta. When it's time to spray, the sprinklers in the farmland will start spraying Penta.
Wow, it's a user-friendly application. With the help of this system, farmers will live a more comfortable life. They can have more leisure time.
Yes, furthermore, we want to release Penta periodically into the farmland in an accurate and most economical way. If we can find how Penta degrades, then it is easy to make a model of simulating the degradation pattern and put the time interval into the device. As UV light has high energy to degrade the Penta, we speculate it is a major reason causing the degradation. Then we model a preliminary function based on the UV light.
And I know you need an experiment to revise it. So we have done experiments, placing peptide under the UV light. We can see the result from SDS page. On the left hand is the original peptide. On the right hand is the result of exposing it to the UV light with intensity approximately 50 milliwatt per square meter and wavelength between 315 to 280 nanometers for 2 hours. As you can see, the remained protein significantly decreased.
So that we can put the prediction modeling on Penta. Finally, we use the function to draw a graph. The x-axis represents the time, and the y-axis represents the amount of Penta. According to this modeling, we suppose that Penta will degrade 20% after 2 days in the environment, so that our device can spray Penta periodically. And we have already written the code into the device. However, before we practically put our system into the practice, we must come to the questions of safety concern. The part still belongs to the biological production. Now, let's Angel continue it for us.
Thank you. First, we have to consider whether it is safe for spraying peptide directly into the environment. In manufacturing our product, we have designed a procedure that guarantees the solution is free of living coli. First, we sonicated the solution of the expressed E. coli, and then we sterilized the sonicated solution by boiling for an hour, which effectively kills off all the living coli in the solution. The SDS page results show the sample boiled in the following timescale. It shows that the proteins didn't denature until the second hour. And also, we apply the boil lyset on the test. The result shows no living coli. Most importantly, the other safety issue we have to consider is peptide itself. As the experiment we showed before, the UV light will degrade pentite over time. For sure.
Wow, it's awesome. But without promoting it, our system can exert its influence.
And next, let us welcome Tiffany to introduce you how we interact with the society.
All the practices can be put into 4 parts: study, education, collaboration, and meetups. In the study, we first consulted the professors in Zhongxin University and Taiwan Agricultural Research Institute for advice about insect cultivations and feeding answers. And we thank Professor Lin Yiping, the inventor of IoT talk system in our school. Also, we visited the tea shop owner and organic farmers in Green Market to ask them about the feasibility and directions of our project. As for education, we held up a 2-week workshop for National Taichung First Senior High School students, and we helped them and Zhongzhou University establish their first team for next year. To investigate the public willingness, we released an online survey. The result shows that 84% of respondents want to try our pentite. Furthermore, we created a board game called Pest Crisis to teach the concept of biobricks for the public. For collaboration, we cooperated with NYMU Taipei to build up a software that can collect long-term weather information and further predict the number of pests. Besides, iGEM team and us help each other confirm the effectiveness of the biobricks, and we provided biobricks to these iGEM teams. Also, we cooperated with other teams to spread an international survey released by Aachen University. For meetups, we're attending many conferences in Asia, and we had online meetups with other iGEM teams to exchange project opinions. What we want to achieve is to solve the problem from the origin, that is, lower the artificial impacts on the environment, and simultaneously adding accurate automation to revamp public stereotypes of agriculture. Let's review what we have achieved in the project. First, for biobrick construction, we have submitted a collection of spider toxins and lectin, a basic part, and a composite part with T-cell promoter and RBS. For the further improvement of pentite, we modified the JS linker of 2010 iGEM NYU team, improved it to be better in expressing pentite. So all in all, we have provided 12 new biobricks in total this year. Second, we have successfully expressed 7 kinds of pentite and proved them to have a repellent effect on lepidopterans. The repellent effect is as good as the existing bioinsecticides Bacillus thuringiensis. Third, we provide a system that is well tailored for the farmers around the world, with an automatic spraying device for sprinkling pentite and a detection device for monitoring the condition in the farmland. In order to prove our concept to be workable in the agricultural industry, we created a product of a complete system that can solve 2 problems in agriculture: food safety and unpredictability. We ask the demands of farmers and public consciousness of related issues. Also, we participated in this agriculture-related startup business competition. Fortunately, we standing out from the crowd with our innovative ideas and thus gaining confidence in their trend.
Here is to a new age of design, a new age of creation that erases the boundaries between biology and engineering. The system is an interplay of hardware and software that finally achieves accurate automation with the application of Internet of Things and sustainable agriculture with the creation of pentite. Now we want to give thanks to all the sponsors, instructors, and all the members in our team for making the project we are today. The system we provide is truly a new choice for farmers that meets the needs and demands for the society. With our system, tomorrow's agriculture is actually today. This is the presentation of NCTU Formosa. Thank you.
Thank you very much for your presentation. As a first question, how did you come up with the idea of using spider toxins? And then also, how did you come up with the idea to improve the system, for example, by adding a longer linker?
For our original brainstorming, we want to solve the problem of using chemical pesticide. So we want to find out a protein that can be used in our project. So we have found scorpions and other kinds of species, and we finally find spiders, because spiders contain in spider venoms, there are a lot of toxin venom peptide that can be used as the potential candidates for the usages in chemical pesticides and biological pesticides. Yes, and for the second question is,
how did you then later made a longer linker? How did you come up with the idea that that would improve performance?
Because our spider venom peptide has the structure, has disulfide bonds, and also lectin is a relatively huge protein. So we think that because our original linker has only 3 amino acids, so we elongate it to 18, and we perceive that it will make them fold better.
In a lot of your data, you have negative and positive controls. Is the positive control a pesticide that's used currently, or is it a plant that wasn't incubated with insects?
The positive control is Bacillus thuringiensis. It's a kind of living bacteria that is found in nature and will form a toxins in their spore and finally kills the pests.
Thank you.
I look at your wiki about your peptide selection procedures, and it seems you put a lot of like filtering criteria. I'm just curious why among them is the organism you chose must be spiders or tarantulas. I'm just curious why would you set that constringent criteria, because there might be peptides from other organisms that also have an insecticide effect.
Yes, for our initial crawling data, we crawl the data from Arachnoserver. It's a spider toxin venom database and also UniProt. And in the UniProt, there are other kinds of toxin peptides that can be used for the potential candidates for our project. But we finally decided to use 3 kinds of spider venom, toxin venoms, and spider venom peptides, because we want to make it maybe uniform. We choose spider venom. And as for other kinds of potential usages, it can be used for future. Thank you.
Hello. I found that you looked at these peptides really interesting, and it was a fantastic talk. So congratulations. So my question is regarding the folding of these peptides, because you got a lot of disulfide bonds, and when you produce proteins, especially non-native to the organism, it's incredibly hard to fold proteins with many disulfide bonds because they can shuffle around. So how do you ensure that the protein, because obviously you show evidence that it works really well, so how do you ensure that these proteins or these peptides are folded well for your application?
Yes, because our proteins also have disulfide bonds, and we have considered it. And first we used a kind of special species of special strains that is called Rositagami for producing disulfide bonds. But actually we cannot ensure that all the proteins we got is in the correct folding ranges. But in the fitting assays, it shows its functions. So we may say that there are a portion of it just in the correct folding, and maybe the other ones is misfold or something. But in the feeding assay, we did see that it has the toxin and those response. Yes.
Do you know what's your best guess regarding to the efficiency of the folding with your modified E. coli that you use, or the modified organisms that allow you to fold the protein? Can you estimate roughly how efficient it is?
You mean the correct folding ones?
Yes, versus incorrect.
Actually, we in our experiment we didn't done that previous. Yes, we haven't done that.
What's your guess?
My guess. Maybe 13 30% or 40%?
All right,
I
thank you
guess.
very much. You highlighted the differences in ion channels in mammals, for example, and insects. But of course, there are many different potential ways that a peptide could affect a human or other agricultural animals. How would you think about trying to ensure it was safe and learning more about that question about whether there were secondary ways that a peptide could affect, or the lectin, for example, could affect humans or agricultural animals?
For these two proteins we use, we have also searched the references that concerning about safety problems, and we all found that the three toxins have been done in the mice test and tested to be not harmful to the mice. And so we perceive that it will not harm mammals. As for, there will be some potential effects that will in either way harm humans. That can be only tested if we are commercializing it turning into a product. We have to do further tests to generally test about it, yes, for commercializing.
All right, a final round of applause for NC2.
Ah oui, d'accord, faut pas le louper. Bon courage.
Alors il est cool leur système, hein.
Il est cool.
Et eux c'est trop cool.
But it looks like and then it's just disturbing.
What are those selfie sticks?
Oh yeah. But it was still like natural. It was still so awesome.
Yeah. I mean, Harvard was good as well, but